Okay, we are continuing with this chapter in muscles. So what we're looking at here is referred to as motor units. Basically, a motor unit is a single neuron and how many muscle cells it is communicating to. So if you look here, motor unit number one is communicating to two muscle cells, whereas motor unit number two looks like to be communicating to three muscle cells. So overall in the body, you got motor units that are small, and then you have others that are large. The advantage of small motor units is that you have better control over them, whereas they don't give you as much force out of the muscle. That would be the disadvantage. And large motor units, well, the advantage is they give you a stronger force, but you don't have as fine control over them as you would the smooth muscle or small motor units, I should say. Let's see here. Let me go to the next slide. So when a muscle contracts, there is going to be a period where the muscle's not contracting, it's not producing any force. So here it's referred to as the latent period. Then as the muscle starts to contract, starts building up a certain amount of force until eventually you get the maximum force out of the muscle. And then after communicating the muscle comes down to relax. And then there's a period of time where the amount of force decreases, where it goes back down to zero. So the force produced by the muscle is referred to as tension. Okay, let me clear this out. Well, some muscles can contract quickly. That would be their advantage, but unfortunately they get tired quicker. So like example, muscles that can contract fast are muscles like in the eye, like the lateral rectus muscle, which moves your eye to the side. So these muscles contract fast, but again, the disadvantage is they fatigue easily. Other muscles, they can sustain a contraction longer. That's their advantage. But the disadvantage of those type of muscles is that takes a while, right? They don't contract as fast, in other words, as muscles like, say, the eye. And then you have some muscles that are somewhere in between. They're not too fast, not too slow. They can resist fatigue somewhat, but not to an extent like muscles that can contract for longer durations. So going back to the muscle called the gastrocnemius, it's your calf muscle. It's somewhat in between. It doesn't contract too fast nor too slow, yet it also doesn't fatigue too easily. So it can sustain a contraction somewhat. There's a period of time where the muscle's going to get excited, and some of these motor units, these are again muscle cells communicated by neurons, start to produce tension, which is the force out of a muscle. And if you can recollect from the last PowerPoint, you need calcium in order for a muscle to contract. So as calcium is being released inside the skeletal muscle, and as long as ATP is available, you're producing tension. And then when you're done communicating to the muscle, the calcium has to go back into the sarcoplasmic reticulum. And as it does that, the muscle's coming back to rest. And then these little arrows indicate the release of neurotransmitters from a nerve cell. So I'm drawing a nerve cell. Here's its axon terminal where it releases the neurotransmitter acetylcholine. You open up, again, chemically gated channels at first. Positive ions with sodium come in, the cells being depolarized. Then eventually, if you create an action potential, you get calcium released to produce tension. Once the muscle is done being communicated to, comes back to rest, the calcium goes back into the sarcoplasmic reticulum. And if, let's say, the neuron releases neurotransmitters again for a second time, you can create the same amount of tension once you create an action potential. And again, when you're done communicating to the muscle, the calcium has to go back into the sarcoplasmic reticulum, and then the muscle's back to rest. So here you're looking at pretty much a neurons communicating to a muscle cell each time you see a little arrow. So let me draw our neuron again. And each time neurotransmitter is being released to create an action potential, it's indicated by an arrow. Okay, so you release neurotransmitters one time, you create an action potential, you get some calcium released, right? Now the muscle's trying to come back to rest, and remember the calcium's trying to go back into the sarcoplasmic reticulum. But before the calcium can be put back, the neurotransmitter is being released again a second time from the neuron. So you don't put all the calcium back. You didn't come back all the way down to rest. So the next time you create an action potential, you're going to release even more calcium. And if you have more calcium released from the sarcoplasmic reticulum, that'll give you more tension, more force produced by the muscle. Again, the muscle's trying to come back to rest, put the calcium back into the sarcoplasmic reticulum. But then the neuron again releases neurotransmitters here for a third time. Doesn't allow the muscle cell to fully come back to rest. Therefore, you don't fully put all the calcium back into the sarcoplasmic reticulum. So even more calcium is released when you can release neurotransmitters within a shorter period of time, giving you even stronger tension. So each time you release neurotransmitters within a short period of time, we say you're increasing the frequency of neurotransmitter release. When you can increase the frequency of your neurotransmitter release, you can get more and more calcium to be released from the sarcoplasmic reticulum, giving you more and more tension, as shown in this buildup effect of the tension. At some point in time, you're almost going to get all the calcium released out of the sarcoplasmic reticulum. So here, the arrows indicate that you're increasing the frequency of neurotransmitter release to get more and more calcium out of the sarcoplasmic reticulum, till eventually you get the maximum amount of calcium released out of the sarcoplasmic reticulum, and then that's going to give you the maximum amount of tension produced by the muscle. So one way to increase the force / tension from a muscle is to increase the frequency of neurotransmitter release so that you can get all the calcium to be released from the sarcoplasmic reticulum. All right, let me jump a little ahead here. This picture is showing us muscle cells as far as size. The size of a muscle cell, of course, will give you stronger tension, but also the motor units. Larger motor units give you larger tension or more force. Small motor units don't give you much force or tension. Again, the advantage of small motor units is that you got better control over them, but they don't give you that much force. So an example would be like muscles in the hand, where you got fine control over those muscles. Okay, let's see, where were we? Make sure I'm recording. So I was just mentioning that smaller motor units, you have better control over them, but they don't give you maximum tension. And when your nervous system communicates to the muscles, the first ones they're going to call in play are the small motor units. Now, if you need more tension out of the muscle, the second size motor units, which are mid-size, will be incorporated next. And then if you need more tension after that, you're going to incorporate the large motor units. So at some point, you're communicating to all the motor units, giving you maximum tension, again, out of a muscle. So another way to get maximum tension out of a muscle, besides increasing the frequency of neurotransmitter release, is to incorporate all the motor units in contraction. And again, the first motor units called in play by your nervous system are the small ones, then the mid-sized ones, until eventually all of them, including the large ones. All right, so going back to this picture, which I just kind of did in the last slide, let me get the pen out. So the first motor units you incorporate are small motor units, indicating by these little circles being colored in. If you need more tension than those, then you incorporate mid-sized motor units as well as the small ones. Then if you need more force, you want to incorporate the large motor units. And now you're communicating to all the motor units together. And eventually that'll give you maximum tension out of the muscle. And that's what this staircase buildup effect of tension is showing. You first incorporate the small motor units, then the mid-sized motor units, and then the large motor units. That'll give you maximum tension from the muscle. And of course, increasing frequency of neurotransmitter release onto the muscle. Okay, this picture here is showing us a contraction. When a muscle contracts, and if it produces enough tension, can overcome the weight that's put onto the muscle. So when a muscle contracts and it overcomes the load and it gets shorter in length, that's referred to as a isotonic contraction. Isotonic. So the graph on below shows us that the muscle is producing force, which is tension, and if it overcomes the load, it'll get shorter in length. Okay, let me erase all this again, or we can go on to our next slide. Now, there are some scenarios where you may have a load put on a muscle, and no matter how much tension you create, you can get maximum tension out of the muscle, but maybe it's not enough to overcome a load. So here you got a muscle. In this graph here, it is showing us that it is trying to overcome the load by producing tension, but it's not enough tension so that therefore its length doesn't get shorter. So think of maybe like a weight. You're trying to pick up a weight and you're producing tension out of your muscle, but no matter how much tension, the maximum tension is not enough to overcome the weight. So again, when a muscle contracts but doesn't overcome the load, it doesn't get shorter in length. That type of contraction is referred to as isometric contraction. Okay, how do cells make ATP? Remember, that's that molecule all cells need to produce in order to survive. You need oxygen to make ATP, and you need a nutrient. The nutrient can be a sugar, or you may use a fatty acid, or even amino acids to make ATP. So the process of making ATP is referred to as cellular respiration. Okay, let's take one of these scenarios at a time. Well, they're not going to break it to us individually. I'm going to have to do it here. Okay, starting with cellular respiration, there's two ways you can go about it, where one is called the aerobic pathway, where you're providing adequate amounts of oxygen to the tissue, and it makes a good amount of ATP. The other pathway is referred to as anaerobic. It's when you don't get adequate amounts of oxygen to the muscle or cell, and you're not making enough ATP. So let's just start off at the beginning. Step one of cellular respiration, you take a nutrient. Let's say the nutrient is glucose. Inside the cell, the liquid matrix is called cytosol. So converting the glucose into what's called pyruvic acid, it happens in the cytosol of the cell. And the process of converting glucose into pyruvic acid, the term is called glycolysis. So again, converting glucose to pyruvic acid happens in the cell cytosol, and it's called glycolysis. And you make a little bit of ATP. Here it says you made a net gain of 2 ATPs. Now, notice that glycolysis, it happens in both pathways. So here you're converting glucose into pyruvic acid. That's called glycolysis. They just didn't label it in the second slide here. So glycolysis happens in both the aerobic and as well the anaerobic pathway, where you make a little bit of ATP. Once you get pyruvic acid created, what happens next will depend if you get adequate amounts of oxygen to the cell. First of all, let's go back to glycolysis. Do you need oxygen to convert glucose to pyruvic acid? This little X mark here indicates that you don't need oxygen to do glycolysis. Okay, so converting glucose into pyruvic acid is called glycolysis. You don't need oxygen. However, what happens next with pyruvic acid will depend if you get adequate amounts of oxygen to the cell or you don't. Okay, let's say you get adequate amounts of oxygen to the cell. The next step is pyruvic acid will enter the mitochondria, and through a bunch of reactions, you make a lot of ATP so that, you could sustain a contraction for a long period of time. Think of doing like a marathon, okay? Well, what happens if you don't get enough oxygen to the cell at this stage? Well, if you don't get enough oxygen to the cell, right here, pyruvic acid will not enter the mitochondria. It gets converted into a waste product called lactic acid. That's pretty much it. The muscle cells fatigue, it's tired. You have to let it recuperate, replenish the amount of oxygen that was used, and then convert that lactic acid back into pyruvic acid. So cellular respiration happens either aerobic or anaerobic. For aerobic, you get adequate amounts of oxygen to the cell. Pyruvic acid can enter the mitochondria, make a lot of ATP. You could sustain an event for hours. The anaerobic pathway, if you don't get adequate amounts of oxygen to the cell, pyruvic acid gets converted into a waste product called lactic acid. This type of making of ATP, you don't make as much, can only sustain a muscle contraction for seconds, anywhere it says from 30 to 40 seconds. Another way skeletal muscle can make ATP is by using a substance referred to as creatine phosphate. Creatine phosphate is a molecule that has phosphate attached to it, and in times when you need to make ATP, will give up the phosphate and add it to what's called adenosine diphosphate. Di means two phosphates. So once you give up the phosphate and you add it to ADP, that'll convert it into ATP. You do not have infinite amounts of creatine phosphate, so making ATP this way will only sustain a short burst of activity. Think of picking up a heavy weight and then dropping it. So here it can only sustain a duration of muscle contraction for about 15 seconds. All right. The other thing I want to bring to your attention here is nutrients. Glucose, which is a sugar, isn't the only type of nutrient you can get from your diet. You may also use lipids. So here you can even use a fatty acid to make ATP, or you can even use proteins. And the monomers of proteins are called amino acids. So you can use amino acids to make ATP. Think of like an assembly line where, let's say, glucose is the first thing at the assembly line to start the process. And then at the end, you make ATP. Fatty acids, amino acids, they also make ATP, but they kind of jump in further along the assembly line. But you're still making ATP. So it looks like fatty acids, amino acids can be used as well to make ATP other than just glucose. All right, so here you got a picture of someone running. It says when they get off the track to run, any ATP they already had in the muscle will be used first. And then what's next? ATP is formed now by using creatine phosphate. So creatine phosphate gives up its phosphate to make ATP. But then again, that can only sustain a short activity, like 15 seconds. Then you start making ATP in a process called glycolysis. You're converting glucose into pyruvic acid. Glycolysis. It looks like you can also use a storage form of glucose called glycogen, which is found inside your skeletal muscle, to start using it as a nutrient. Okay, glycogen is the storage form of glucose. Now, after glycolysis, that's not enough ATP to sustain an event for hours. However, if you do provide adequate amounts of oxygen to the muscle cell, you can make ATP what's called aerobically, and you make a lot of it, and you can sustain an event for hours. If you do not get enough oxygen to the muscle cell, after glycolysis, that's it. The muscle cell, they're tired, they're fatigued. You gotta allow them to recuperate. Think of like lifting weights and you feel the buildup of what's called lactic acid. You'll feel the burn in your muscles to a point where your muscles can't contract anymore. You have to put down the weight, allow the muscle to recuperate by replenishing oxygen that was used and converting the lactic acid back into pyruvic acid. Okay, let me clear out these drawings. Oops, that was not what I wanted. So just give me 1 minute. Okay, I believe we're recording again. Just make sure. Yes. Okay, this picture here is just showing us again, how can we get a stronger or more tension out of a muscle? One, we had mentioned to increase the frequency of neurotransmitter release. When you increase the frequency of neurotransmitter release, you get more and more calcium release to eventually give you maximum tension out of the muscle. So here, increase the frequency. It says stimulation. They're referring to the release of neurotransmitters in this case. Time. What are you affecting? You're shortening up the time between neurotransmitter release. In other words, you're increasing the frequency of its release. The other way to get stronger force out of a muscle is to communicate to large motor units to eventually communicate to all the motor units. So large motor units and then all of them together give you a maximum tension. The third or the last thing here is the length of a muscle. There's a certain length of a muscle that will give you the best amount of tension. So let me clear the drawings. So let's look at the sarcomere. Remember, the sarcomere is the functional unit of a skeletal muscle. It is an area from one Z disc to another Z disc. Now, myosin in red here is the protein that likes to bind to actin in blue. In order for myosin to bind to actin, the actin has to be directly above the myosin heads. So in the first scenario, this muscle is already almost fully contracted. It's not going to give you much more tension. In the far right scenario, the muscles overly stretch where myosin is not overlapped any longer by actin. It's not going to give you a strong tension. Therefore, the best scenario is when myosin is directly over the, I should say, when actin is directly over the myosin, that'll give you the best tension. So there's a certain length, anywhere it says from 80 to 120% of the muscle's resting length will give you better tension. All right, comparing the types of muscles. Muscles that some of them we mentioned were fast, but they get tired easily. So muscles that are fast, they're referred to as fast glycolytic fibers. So yes, their speed is fast. However, they get quite fatigued easily. Slow oxidative, okay? These muscles, unfortunately, yeah, they're slow, but look, they can resist fatigue long term. And then the fast oxidative, those are the intermediates between the two. They're not too fast, nor are they too slow. All right, what are some other things you can compare? How do they make their ATP? Oxidative. That means that the slow oxidative are making their ATP aerobically. It means you're getting enough oxygen to the cell. Therefore, you don't get tired as easily, and you could sustain an activity for up to hours. Think of maybe like a marathon. Whereas the fast glycolytic, they're fast, but they're totally dependent on glycolysis. So that means they're making their ATP anaerobically, which means they don't make too much ATP. So they get fast, tired easily, but that can only sustain an activity short term. Okay, so think of like hitting a baseball. And of course, the intermediate is just something in between the two. You're not going to sustain a long-term event or necessarily a short term. It's somewhat in the middle. Okay, myoglobin. Myoglobin is a protein pigment similar to hemoglobin in that it can hold oxygen reserves for muscle in times when it needs to make ATP. So myoglobin, if you find more of it in a muscle, gives the appearance of red. So slow oxidative muscles or slow oxidative fibers, they have high amounts of myoglobin. So they look more red in color. Fast glycolytic, well, there's not much myoglobin in the muscle, so they look more white in color. And then the intermediate, well, that's going to look what? Somewhat pink, the in between white and red. Another thing we can compare is the amount of mitochondria. Remember, mitochondria are organelles that make ATP. So because slow oxidative can resist fatigue and they make their ATP aerobically, it makes sense that they would have a lot of mitochondria so that they can resist fatigue by making high amounts of ATP. Fast glycolytic, they get tired easily. They make their ATP anaerobically, totally dependent on what's called glycolysis. So that means they don't have too much mitochondria. They have very few. And another thing you can compare are capillaries. Capillaries are the blood vessels that are the smallest where gas and nutrient exchange is going to occur. You're delivering oxygen, nutrients to a tissue. So those slow oxidative fibers, they're slow, but through capillaries, you have a lot of them delivering oxygen and nutrients to the muscle cells so that you can make your ATP aerobically. The fast glycolytic, well, they don't have that many mitochondria, okay? So they have few. And actually, I should have done this backwards here. But anyway, they have few mitochondria. And because they have few mitochondria, they don't make abundant amounts of ATP. So they're going to get fastly tired. It's getting a little messy. Let me clear this up. And let me see here. Glycogen. Glycogen is a storage form of glucose. So anytime you have extra glucose, your body hooks it up, similar to hooking up boxes of a train, and storage in a form called glycogen. You can store glycogen in your skeletal muscles, but also your liver. So let's see which one is more or has more glycogen in-house to make ATP. So let's go with the glycogen. The fast glycolytic have the highest amount of glycogen, so it's more easily available. If something's already available inside the muscle, it can use it quickly. Therefore, it can contract faster. The slow oxidative, they don't have that much in-house storage of glycogen, so they do not contract as quickly. They're more dependent on getting their nutrients from capillaries, which takes a while. And then last, the diameter. Which ones are the largest in size? It looks to be the fast glycolytic, whereas smaller in size are the slow oxidative. The percentage of the slow oxidative and fast glycolytic in the body depends mainly on your genetics, how much you have percentage-wise of each. When you train, to do an event, you can try to get the fast glycolytic to look like an intermediate. How do you do that? Well, when you train, you're going to be producing more capillaries to provide oxygen and nutrients to a muscle. When you train, the muscle will get larger in diameter. When you train, what else? We already said that you make more mitochondria as well. Okay, so you can get these fast glycolytic through training to look somewhat like an intermediate, but you cannot, and I stopped sharing again. Let me get that back. Make sure I'm recording, but you cannot get the slow oxidative fibers to look like the fast glycolytic. So again, training wise, you can get fast glycolytic to look like an intermediate, but you cannot convert slow oxidative to look like an intermediate. All right, let me erase all this and go to the next slide. Okay, what are some things to help you to sustain a contraction of a muscle and yet do it quickly? Well, it helps if the load is very light, right? You can sustain a light load for the muscle to contract over a long period of time versus a heavy load. Okay. The other thing here is that you can communicate to sustain a contraction longer, what's called the slow oxidative fibers. They make their ATP aerobically. If you want a fast contraction, well, then that's when you're going to communicate to the fast glycolytic fibers. Now we're looking at smooth muscle, okay? Skeletal muscle you find attached to your bones. Smooth muscle you're going to find within the walls of organs, pushing a solid or a liquid. Now, most often that's where you find it, within walls of organs, pushing a solid or liquid. So here, like in digestive organs, you're going to find smooth muscle to help push solids. When you look at smooth muscle, it looks a little bit different than the point of communication of a neuron and the muscle versus skeletal muscle. Doesn't quite look the same, but you're still going to be releasing neurotransmitters. So you got these little, what's called varicosities at the end of the axon where the neurotransmitter is going to be released. Smooth muscles tend to taper, which means they come to a fine point at their ends. Smooth muscles are non-striated, so you don't see the stripe appearances like you would in a skeletal muscle. Smooth muscle has also these little infoldings here, and they are called, well, let me see, not gap junctions, they're just little infoldings of the cell membrane. And then they have gap junctions, okay? The little infoldings are to allow from calcium to come into the muscle cell. For skeletal muscle, calcium is solely coming from the inside of the muscle, which was stored in an organelle called the sarcoplasmic reticulum. But yet for smooth muscle, some of the calcium comes from outside and enters in these little infoldings inside the smooth muscle. Smooth muscles also have proteins called gap junctions that will connect muscle cells together so that when excitation spreads into the muscle through gap junctions, it can quickly spread to the next muscle, and that causes muscles to contract together rhythmically. The other thing is smooth muscle, you do not have voluntarily control over the smooth muscle like you would as skeletal muscle. Smooth muscles only have one nuclei per cell. If you recollect, skeletal had many. Smooth muscle also have these proteins called intermediate filaments that will allow the muscle to recoil after it contracts. So this picture here is showing us that for a smooth muscle, some of the calcium is not just coming from its sarcoplasmic reticulum inside the muscle cell, but some of the calcium is coming from outside the cell and entering through these little infoldings called caveolae. So that means the smooth muscles, sarcoplasmic reticulum, is not as extensively developed as you'd find in a skeletal muscle. So calcium comes from outside as well as from the sarcoplasmic reticulum for a smooth muscle. The other difference here as far as how's the calcium being used in the smooth muscle? Well, in the smooth muscle, in this drawing, you're looking at myosin, and above the myosin heads in blue are actin. For skeletal muscle, the binding sites on actin were covered up by a protein called tropomyosin, and you needed calcium to expose binding sites on the actin. But here, if you look in the smooth muscle, the binding sites where myosin likes to bind to, they're not covered up. So that means calcium is being used a different way for a smooth muscle contraction. So how's the calcium being used? Well, calcium actually, it activates a molecule, protein called calmodulin. When calmodulin is activated, it activates now enzymes called kinases. After kinases are activated, those enzymes called kinases will help shuttle phosphates, the P phosphates from ATP, and help shuttle it to the myosin head so that the muscle of a smooth muscle will contract. So kind of put it all together here. Calcium not only comes from the sarcoplasmic reticulum inside a smooth muscle, but also is coming from outside the cell, entering through infoldings called calveolae. Calcium will next activate a molecule called calmodulin. Calmodulin will activate enzymes called kinases. Kinases will help shuttle phosphates from ATP to the myosin heads, therefore allowing the muscle to contract. This picture is showing us again that skeletal muscle, if you can remember, is multinucleated. There's many nuclei per cell. So at some point when you're developing as a embryo slash fetus, you had single cells eventually fuse together to make one long skeletal muscle cell. Therefore, that explains why you see that one skeletal muscle has many nuclei. We say it's multinucleated. All right, let's have a look here. See if I can magnify. No, probably not. So, looking at the skeletal muscle, kind of just a little review. The outer connective tissue of a skeletal muscle is referred to as epimysium. The units pulled out of the skeletal muscle are referred to as fascicles. And then the connective tissue around the fascicle was referred to as perimysium. And then the units inside the fascicle, pull one out, is a muscle fiber, which is a muscle cell. The connective tissue around the muscle cell is called the endomysium. The cell membrane, however, the muscle cell is called sarcolemma. The units, if you were to pull a unit out of a muscle cell, is called a myofibril. The myofibril consists of thick proteins in red called myosin, thin ones in blue called actin. They overlap each other. It looks darker, is referred to as the A band. If you only see actin, it looks appearance lighter, it's referred to as an I band. Okay, we did this chart earlier. It's just a different color. There, in this picture, we want us to compare and contrast the three muscle types. Well, the three muscle types are skeletal, cardiac, and smooth. Each muscle is going to provide for some type of movement. For skeletal muscle, it's for voluntary movement, It's the only one that you're consciously aware of controlling. It's for communication. And in times where you're cold, it'll contract quickly to produce heat. So that is the function of skeletal muscle. Where do you find it? Attached to your bones. Cardiac muscle, you find it within the walls of the heart. The function is to propel or push blood. Smooth muscle, most often you're going to find it within the walls of an organ where you're pushing a solid or a liquid through the organ. Another thing you can compare, or let's say contrast, are the number of nuclei. For skeletal muscle, there's many nuclei per cell, so we refer to it as multinucleated. For cardiac muscle, you can have up to 1 nuclei per cell or 2, which means binucleate. For smooth muscle, typically you only have 1 nuclei per cell. What are some features you may see in one muscle or not in another? So for skeletal muscle, you see dark regions and light regions called striations. So they do have them. Cardiac muscle, you also have striations. What causes the striations are the arrangement of the proteins, myosin and actin. If myosin overlaps actin, it looks darker. That's your A band. If it looks lighter, where there's only actin, that's looking lighter, it's an I band. So you have striations in both skeletal and cardiac. You don't see striations in smooth muscle. For smooth muscle, it tends to taper, come to a fine point at its ends. For skeletal muscle, it's very long, cylindrical. Cardiac muscle tends to branch out. The other thing here is cardiac muscle has special proteins called intermediate filaments that will allow for excitation to spread from one cell to the next so that the cardiac muscle can contract rhythmically together. Some other things I'll bring to your attention: when you work out skeletal muscle, it can get larger in size by adding protein, more protein. That, when it gets larger in size by adding more protein, is referred to as hypertrophy. Cardiac muscle as well can hypertrophy. Smooth muscle does not get larger in size through adding more protein. Typically, it can replicate, like in the uterus, to propel baby for birth. And the term for it to get larger in size is referred to as hyperplasia. So smooth muscle does hyperplasia, whereas skeletal and cardiac do what's referred to as hypertrophy. By adding protein, they get larger in size. The other thing is, if you don't use a muscle, it'll degenerate. We say, well, atrophy, degenerate, get smaller in size. You may see someone with a spinal cord injury. They lose voluntary communication to their muscles. It'll degenerate. It'll atrophy, we say. All right, comparing the three muscles, you can see you have the outer connective tissues, epimysium, perimysium, endomysium, and skeletal muscle, whereas cardiac, you only see endomysium. For smooth, you only see endomysium. Is there a presence of myofibrils composed of sarcomere? Sarcomere, again, is the functional unit from one Z disc to another Z disc. So for skeletal, yes, you have these sarcomere regions from one Z disc to another Z disc. Same for cardiac. For smooth, it says no, but you still have the proteins, actin and myosin, involved for the muscle to contract. Which one has the most developed sarcoplasmic reticulum? So it doesn't have to rely on calcium from outside the muscle cell. That would be skeletal muscle. The next well-developed sarcoplasmic reticulum would then be cardiac. The least developed would be smooth. So some smooth muscles, they only have infoldings to get their calcium from outside the cell, called calveolae. What are we looking here? We're looking at the point of communication. What can communicate to the muscle? So all three muscle types can be communicated through your nervous system by the release of neurotransmitters. Just remember, the only one that you can voluntarily control that you're aware of is skeletal muscle. Which one of these muscles have these proteins called gap junctions to allow excitation to spread from one cell to the next? Definitely for cardiac, since the heart contracts rhythmically together, you'll have what's called gap junctions. For smooth, just depends where the smooth muscle is located. If you're in digestive organs, you're going to see the proteins called gap junctions. Which one of these muscles besides the nervous system can be communicated by hormones like adrenaline? Well, definitely the cardiac muscle, right? You can communicate through hormones. Different hormones as well can cause smooth muscles to contract. Which ones can contract when being stretched? Well, typically cardiac and smooth muscle. So to give you an example, think of smooth muscle in your digestive organs. So when contents enter the digestive organs, that stretches the muscle and it initiates a reflex for it to contract. So simply stretching either smooth muscle or cardiac muscle will cause it to contract. Whereas skeletal muscle, the only way it'll contract is to communicate by releasing neurotransmitters. Again, how's the calcium used for each muscle to contract? Both for skeletal and cardiac, calcium is used to expose the binding sites on actin so that myosin can bind to actin. As for smooth, calcium is used a different way. It activates a molecule called calmodulin, which will eventually activate enzymes to help shuttle phosphates for the muscle to contract. Pacemaker cells are type of cells that are not involved in a contraction but can excite the muscles around it. So you may have seen documentaries or surgeries of the heart where it's out of the body and it's contracting on its own. That's because of pacemaker cells. So does skeletal muscle have pacemaker cells? No. However, you do have pacemaker cells for cardiac muscle and areas of muscle where you're pushing a solid or liquid, like your digestive organs. You may as well see gap junctions and pacemaker cells. Pacemaker cells set the excitation, set muscles contract rhythmically together. Now, some muscles, they can be excited by releasing neurotransmitters, but also they may be inhibited by release of neurotransmitters. For skeletal muscle, when you release neurotransmitters, you can only excite it. If you don't excite it, well, then it's at rest. For cardiac muscle, you can release neurotransmitters to excite it or to tell it to rest. Don't contract. Same thing for smooth. There's some type of neurotransmitters that may excite or as well inhibit. Which ones muscles that will contract rhythmically together? Most definitely cardiac muscle through gap junctions. And in areas of digestion, will have rhythmic contractions so that muscles contract together. Which ones can contract in response to being stretched? That'd be again cardiac and smooth. Which one here does make its ATP aerobic or anaerobically? Well, let's see. For skeletal, it can do either or. It can do aerobic to sustain contractions long term. However, if you don't get enough oxygen to the muscle cell, it may produce its ATP anaerobically. For cardiac, you never want it to stop, right? So it's got to produce its ATP aerobically and as well have the most abundant mitochondria, so it will not rest. For smooth muscle, it tells us mainly it contracts through aerobic cellular respiration. Kind of just review real quick. Here you're showing a neuron communicating to a muscle, the point of communication between the muscle and the neuron. It's called a neuromuscular junction. At the end of the axon, this round bulbous region is called the axon terminal. Then you have these vesicles that contain the neurotransmitter. They're referred to as synaptic vesicles. There's always a little space between the neuron and the target. So a little infolding on the cell membrane of the muscle cell. It's referred to as the motor end plate. Okay. And again, the axon never physically touches the target. It's always a little space called the synaptic cleft, but the infolding altogether is the motor end plate. The first type of channels that open up in response to acetylcholine are chemical gated channels. At some point in time, the neurotransmitter has to come off so that the muscle can come back to rest, or you'll see enzymes break down the neurotransmitter. Once you open up voltage-gated channels, and if you change the voltage to a certain threshold, the next type of channels that will open up in response for sodium are called voltage-gated. If I open up one, like dominoes all falling, in this case channels, they'll all open, and that causes the release of calcium. For a skeletal muscle, calcium will expose the binding sites on actin. And all you need is ATP, and if the binding sites are exposed on actin, myosin can bind to it. And then the ATP provides energy to do what's referred to as the power stroke. Now, we did mention ATP. What happens if you pass on? You're no longer making ATP. So notice ATP disattaches the myosin from actin. Attachment of myosin to actin is called a cross bridge. So yes, we need ATP to disattach any cross bridges. See it come off the actin on the bottom picture. Once ATP gets hydrolyzed, that provides energy to pull back this hinge on the myosin. And then calcium is available. That means the binding sites are exposed so that myosin can bind to actin. And when the ADP and phosphate leave myosin, that will provide the energy to push the myosin and actin toward the middle. And if you do this on both sides of the Z disc of a sarcomere, the whole sarcomere will get shorter in length. Do enough sarcomeres in the muscle, the whole muscle will look as if it's getting shorter in length. Now, when you pass on, you no longer make ATP, so any cross bridges attach, there's no ATP present, the myosin's going to stay attached and give the body condition where it looks rigid called rigor mortis. The only way rigor mortis is going to dissipate or to stop is through natural time, where the proteins will be eventually broken down, where the myosin is attached to actin. Over time, you can break the protein down. And that's the end of this chapter, and I will see you next time. We'll continue on, I believe, in chapter 11. I'll have to make sure. But anyway, until then, I'll talk to you then.